Good morning. My topic today is why One Health is critical for public health. I'm Malik Peiris at the School of Public Health, and I have no conflicts to declare. As we all know, we are going through the traumas of COVID-19 with huge impact in terms of human cases, hospitalization, deaths, and a massive impact to the economy. And we know that this particular pandemic came to humans from animals, the reservoir being rhinolophus bats. We don't know exactly how it got to humans, but it certainly came from these rhinolophus bat species. Now, of course, over the last 20, 30 years, there have been many emerging infections that have crossed to humans. And most of these are zoonosis, meaning that they come to humans from animals. And most of these are caused by RNA viruses because RNA viruses have a high mutation rate, which means they have a greater capacity to adapt when they cross species from one species to another. Now, this illustrates the pathways of emergence of these viruses. We have huge diversity of viruses out in wildlife. We have viruses endemic in animals. Domestic animals. Sometimes these wildlife animals cross over to domestic animals or either directly cross to humans or through amplifier hosts or domestic animals cross to humans and may cause zoonotic disease. This can be sporadic, maybe very just one case or a few cases. And examples of this are avian influenza H5N1, H7N9, MERS, Nipah, Ebola, etc. But very occasionally, these spillovers become established with efficient human-to-human transmission. And then the epidemic or pandemic begins to spread regionally and globally. So influenza pandemics behave like this. SARS coronavirus 1 in 2003, COVID-19, and then two of the coronaviruses that are now endemic in humans, 229E and OC43, which caused the common cold, we know now that they also jumped from animals to humans within the last 300 or 400 years. Now, some of these viruses, after getting established in humans, also can go in the reverse direction, back to animals. So, for example, the pandemic H1N1, which came from swine to humans, as it spread worldwide, it has gone back into swine and got established in swine and is reassorting with swine influenza viruses. Similarly, I'm sure you're aware that SARS coronavirus has spilled back into pet species such as dogs, cats, but also into farmed animals such as mink, and can cause major problems there as well. Now, the factors that lead to the emergence of these new viral infections are changes in animal production. And here you can see the very dramatic increase in dairy products, in animal products, meat products over the decades. International travel, a huge increase in international travel, so that any outbreak that takes place anywhere in the world today can be in any other part of the world within 24 hours. Ecological changes, deforestation, climate change, the pet trade, changes in human behavior, all these contribute to emergence of new virus infections. So this means that when we are dealing with these emerging infections, we really have to deal with it not just from the human health sector, but with the animal health sector and the environmental sciences together. And the definition of One Health is trying to attain optimal health for people, animals, and the environment. So this is a schematic of how these spillovers occur. As I said, these viruses may be present in wildlife. They may spill over to domestic animals and from there spill over to humans, or directly spill over from wildlife to humans. And there are at least 260 viruses in 25 virus families that are known to infect humans. Now, what we are trying to do is to reduce the risk of these spillovers. So, for example, reduce the risk of spillover from wildlife to animals and from wildlife to humans, and also the spillovers from domestic animals to humans. So for this, we need the surveillance in humans and in animals. So again, the need for One Health. We need to risk assess these potential spillovers, for example, with zoonotic influenzas, which of them might become potentially pandemic. We need to try to understand how these spillovers occur so that we can reduce the risk of these spillovers taking place. And then, of course, finally, once a human outbreak takes place, early detection and response may reduce the scale of the epidemic. Now, in terms of pandemic preparedness, we need, as I said, early detection and characterization of human outbreaks. And this is to calibrate your response, the containment mitigation policies, to understand disease severity, and to understand routes of transmission, so you can reduce the chance of these spillovers and also transmission in humans. And of course, in this regard, after the 2003 SARS outbreak, the international health regulations came into being. Then, of course, there is what I call getting ahead of the curve. So this is the 2009 influenza pandemic. We heard about it in April of 2009, about a new pandemic virus in North America. And this data shows the infection attack rate in the population in Hong Kong. And these are children. You can see that by September, October, almost 40% of children in Hong Kong had been infected with this new virus. So waiting until a pandemic starts is too late to activate interventions, vaccines, and antivirals. So you have to get ahead of the curve. And in order to do this, one needs to identify potential threats and risk assess which of these viruses may be potentially zoonotic and pandemic, so that we can develop countermeasures to these. So, for example, MERS coronavirus, the coronaviruses we find in bats, avian influenza viruses, swine influenza viruses, Nipah. These are some of the threats that we can clearly see out there that we need to be developing countermeasures to. And in order to identify these threats, we need surveillance at the animal-human interface. Again, One Health. And it's not just doing surveillance and detecting viruses, because there are literally thousands or tens of thousands of viruses out there. We have to try to risk assess which of these are likely to have the capacity to establish themselves in humans. And it's important to point out that in reality, we had advance warning that SARS coronavirus-like viruses were significant threats to humans. And indeed, we also had early potential warning of the 2009 pandemic. And what we failed to do was, in those cases, not just surveillance, a lack of surveillance, but a lack of risk assessment and also action to develop countermeasures. So if I take the example of influenza, we know that influenza viruses in humans ultimately come from animals, which establish themselves in humans as pandemic viruses and then become seasonal influenza viruses. Now, in order to assess or risk assess these zoonotic viruses such as H5N1, H7N9, H5N6, etc., that spill over and cause zoonotic disease in humans, over the last decade or so, the WHO and also the US CDC have established a systematic approach to assessing risk. And this is called the Tool for Pandemic Risk Assessment by WHO, TIPRA, or IRAT by US CDC. And the idea with this is to prioritize preemptive development of vaccine candidate strains. So you can see it's not only a matter of detecting the virus, there's a whole range of other parameters that go into this risk assessment. the immunity in the population, the receptor binding properties of the virus, transmission in experimental models, of course, the sequence of the virus, etc. So it's quite a range of parameters that we have to get information on to make this type of risk assessment. And then the assessment is made in 2 dimensions. One is the risk of emergence, the chance that a virus may emerge to become transmissible in humans. And the other is the impact risk, meaning if such a virus becomes transmissible, how severe would it be? So obviously, if you have a virus that is at high risk of emerging in the human population, but is very low severity, then the risk is relatively modest, if you want to put it that way, even if it does emerge in the human population. On the other hand, if you have a virus that has a high emergence risk, but also high impact risk, say, for example, H7N9, then those are the types of viruses we have to be concerned about and make vaccine candidate strains too. Right, now, this type of risk assessment strategy has been fairly well developed for influenza, but not for other groups of viruses, such as coronaviruses or others. But now, more recently, just a few months ago, there was an attempt to try to risk assess some of these other virus groups as well. And also, there are studies, for example, trying to assess the spillover risk of SARS-related bat coronaviruses in Southeast Asia. And in this particular preprint, it is not yet accepted for publication. They have estimated that about 400,000 people are infected every year with SARS-like coronaviruses in South and Southeast Asia. Now, this is not COVID-19. These are other bat SARS-like coronaviruses. So these are certainly a group of viruses that pose further pandemic threats. Now, that's getting ahead of the curve. Then there is the importance of trying to control at source. Now, what do I mean by that? What I mean is, by understanding the pathways of a virus spilling over to humans, one can take interventions that reduce the risk of these spillovers, so that even without necessarily identifying which virus is going to cause the next epidemic or pandemic, you can generically reduce the risk of these spillovers taking place for different groups of viruses. So if you take the example of avian influenza viruses, such as H5N1 or H7N9, we, over the years, have clearly established that what happens is that these viruses get amplified along the poultry marketing chain, and once they enter these retail live poultry markets, which are found in many countries in Asia, the virus never leaves. So, because you always have new poultry coming in and poultry are being slaughtered. And the evidence for that comes from studies we did in Hong Kong, where with a low pathogenic avian influenza virus H9N2, we were able to show that these are now here. We have 6 live poultry markets here, and we closed the market for 1 day. And this is before the closure. You can see that 5 of these markets have evidence of virus, maybe about 5% of poultry carrying virus. But after closing the market for 1 day and then reopening, and now poultry coming back in, you can see the rates of infection have dramatically reduced, close to 0. But by next month, it is up again to 5, 6, 7%. Again, closure, it goes down, but then up again. So what it means is that the only way to break this chain of transmission in the live poultry market is completely empty the market of poultry. And also, studies show that when there is outbreaks in poultry farms, this is associated with the virus being moved from retail poultry markets, where they're amplifying, through cages and people movement back to poultry farms. And this is studies we have done over 12 years in the live poultry markets in Hong Kong. And you can see the impact of these interventions progressively reducing the carriage of avian influenza viruses. And then finally, with the introduction of banning of keeping poultry overnight in the market, you can see almost complete abolition of virus persistence in these markets. So after 1997, really with these measures that have been introduced in Hong Kong, we have had no further cases of severe avian influenza disease acquired within Hong Kong. And we have also gone on to do other studies, and this is done in southern China in collaboration with our colleagues in Guangdong, where we showed that the virus can be found in air. Infectious virus can be found in air in the vicinity of these live poultry markets. And we also able to show that the highest concentration of virus is around these defeathering machines, where once the poultry are killed, they are put into this, and this spins around quite rapidly to defeather the birds, but it then leads to creation of aerosols. And with this knowledge, we have now worked with our engineering colleagues to make minor modifications to these defeathering machines to reduce the spread of aerosols from these defeathering machines. Then also, we can understand through evolution studies how these zoonotic influenza viruses emerge. So, for example, we know that with the H7N9 virus, basically influenza viruses have 8 gene segments. Now, 6 of these gene segments came from a chicken virus called H9N2, but then with the H7 and the H7 and the N9 coming from wild bird viruses, and these wild bird viruses first spill over into domestic ducks, and because domestic ducks and chicken and quail and other terrestrial birds are marketed together in these live poultry markets, you have ample chance of mixing of these viruses and reassortment taking place. And that's the pathway of emergence. And this is not just true for H7N9, but also for H5N1 and other zoonotic viruses. So by separating the path, if we can separate the marketing of these aquatic poultry from terrestrial poultry, we have a generic way of reducing risk of the emergence of these zoonotic viruses for the future. Now, of course, with SARS in 2003, we know that virus got to humans through exposure to viruses in these wild game animal markets, not live poultry markets, but these are wild game animal markets, and animals such as civet cats and raccoon dogs and such like. And with that information, those markets were closed later in 2003, and that closure almost certainly prevented the virus spilling back into humans after the human outbreak had been stopped. Subsequently, of course, we know that the natural reservoir is these rhinolophus bats, but the spillover to humans occurred in the case of SARS in 2003, not directly from bats, but through spilling over into these intermediate mammalian hosts, such as civet cats, which are kept in these game animal markets. And the virus, again, like the live poultry market setting that I was telling you about, once the virus gets established in these game animal markets, it continues to stay there, and humans continually get exposed until finally a virus adapts to acquire efficient human-to-human transmission. Now, finally, I want to talk about MERS coronavirus. I'm sure you know about the Middle East respiratory syndrome. Now, all human cases have occurred basically, the zoonotic infections from animals to humans have occurred in the Arabian Peninsula. And we know that the source of human infection are these dromedary camels. From time to time, of course, infected humans from the Middle East have traveled to other parts of the world and have started outbreaks or cases in other parts of the world. But the zoonotic spillover has basically occurred in the Arabian Peninsula. But we know that dromedary camels, the vast majority of dromedary camels are found in Africa. almost 80% of the global population is found in Africa. Only 4% of the global population of dromedary camels are found in the Arabian Peninsula. And wherever you find these dromedary camels, there is evidence of MERS coronavirus, with a few exceptions, such as Kazakhstan and Australia, for example. So the question is, why has zoonotic MERS in humans not being detected in Africa? So this is one of the questions we wanted to try to address. And first and foremost, we did studies in a number of countries in Africa, in Morocco, Egypt, Ethiopia, Kenya, Nigeria, Burkina Faso. So this is East Africa, North Africa, West Africa. And we were uniformly able to show that the virus activity in these camels is exactly similar to what we see in the Arabian Peninsula. Then we also were able to show that the extent of exposure of humans was no different. But when we did antibody studies, or when we took blood from camel workers, there was little or no serological evidence of infection in these camel workers. But if we do T-cell studies, we can show evidence of specific T-cell responses to MERS coronavirus. So in a study that we did in Nigeria, about 1/3 of these camel abattoir workers had evidence of specific T-cell responses to MERS coronavirus. So that means there is human infection taking place. But why is there no human disease being reported? Now, when we look at these viruses from Africa, we find that they are genetically distinct from those in the Arabian Peninsula. And we then phenotype these viruses by characterizing them in cell culture and for their growth properties in ex vivo cultures of the human lung. And we were able to show that the viruses from Ethiopia, Kenya, Egypt, Nigeria, Burkina Faso, and Morocco, all of them have lower replication competence in the human lungs. And we can show that it is the spike protein of MERS coronavirus that contributes to this biological phenotype of lower replication competence. But the spike protein of the African MERS viruses differs only in 6 to 8 amino acids from the more pathogenic viruses that are found in the Arabian Peninsula. So we would caution that we really need enhanced surveillance of MERS coronavirus at the animal-human interface across the dromedary camel herding regions in Africa. Because as I showed you, we do think that infection is taking place, and we really need to learn much more about what is exactly going on with MERS in Africa, because, as I said, almost 80% of the source of infection, which are these infected dromedary camels, are found in Africa. Now, then finally, the final option in terms of pandemic preparedness is enhancing our response capabilities for countermeasure development, and that is vaccines, antivirals, and diagnostics. Now, we cannot simply rely on the pharmaceutical industry to do this, because who in the pharmaceutical world is going to put in money to develop a vaccine for a virus that there is no certainty there will be a need for a vaccine? Because some of these viruses, if you take MERS, the extent of human infection at the moment is quite low, and there is no major market to cover the development cost for such a vaccine. So this, we really need alternative funding strategies for the development of these vaccines, and these fundings have to come from the government sector. For example, we have CEPI, the Coalition of Epidemic Preparedness Initiative, which has been doing a great job with trying to fund the development of vaccines and antivirals for some of the high-threat pathogens. With that, I will end. Thank you very much for your attention. Thank you.